Hello and a massive welcome to all of Mr. Cheese students. I'm your host, and today we're kicking off our mission to absolutely dominate the October/November 2024 Cambridge Biology Paper 1. We're going to start by breaking down the first 10 questions, showing you all the tricks and strategies to nail every single one.
So think of this explainer as your secret weapon, your personal coach walking you through every step. We're here to build up your confidence so you can walk into that exam and smash it. All right, grab your notes, get in the zone, and let's get started.
Okay, let's dive right into question 1. This is a classic magnification problem. We've got a diagram of a blood capillary, and they've given us a key piece of information. The actual size is 7 micrometers. Our job? Find the magnification. First things first, you got to grab a ruler and measure the image. On this paper, the diameter of that lumen measures up to exactly 35 millimeters.
Now listen up, because this is where the exam loves to set a trap. The formula is easy. Magnification equals image size divided by actual size. But here's the golden rule you absolutely can't forget. Make sure your units are the same. Our image is 35 times millimeters, but the actual size is in micrometers. So what do? We convert. We multiply 35 by 1,000 to get it into micrometers. That gives us 35,000 micrometers. Now we can divide. 35,000 divided by 7, and boom, that's 5,000. See? That unit conversion is everything. It locks in D as our answer.
All right, from microscopes, we're zooming way in for question 2. It asks, which statement explains why lymphocytes with no nucleoli die? This is all about connecting the dots, linking this tiny little structure, the nucleolus, to its massive life or death job in the cell. So here's the logic step by step. Think of the nucleolus as the ribosome factory. And what are ribosomes? They're the protein-making machines of the cell. So if you don't have a factory, you can't make any machines. If you don't have any machines, you can't build anything. No proteins, no enzymes, nothing. The whole cell just grinds to a halt, and it's game over. For the high flyers, you'll spot this right away. The other options are just red herrings. The real issue, the bottleneck, is the lack of protein synthesis. That makes D the only possible answer.
On to question 3. This one's like a cellular sorting game using a technique called density gradient centrifugation. Just imagine shaking a jar with rocks, pebbles, and sand. What happens? The heaviest stuff, the rocks, sink to the bottom, right? It's the exact same idea here, but with organelles. And watch out, the question asks for the order starting from the top of the tube. So we're looking for the lightest stuff first.
So let's break it down. Who's the heavyweight champion of the cell? That's the nucleus for sure, so it sinks right to the very bottom. Next up, a bit lighter, are the chloroplasts. Then come the mitochondria. And floating at the very top, like tiny little bits of dust, are the ribosomes. But remember, the question wants the order from the top down, so we just read our list in reverse. Ribosomes, then mitochondria, then chloroplasts, and finally, at the very bottom, the nucleus. That sequence lines up perfectly with option D.
Question 4 is a classic knowledge check, but you have to be careful. It's asking which cell structures contain nucleic acids. This isn't just about the nucleus. You got to think a little broader and check every single suspect they give you.
Okay, let's go through the lineup. Chloroplasts? Yep, they've got their own little circle of DNA. Golgi bodies and lysosomes? Nope, nothing there. Now what about ribosomes? This is the one that sometimes tricks people. But yes, absolutely. Ribosomes are literally made of ribosomal RNA, rRNA, which is a nucleic acid. So our two structures are number 1 and number 4. That means the right combination is C. Case closed.
Now for question 5, a sequencing puzzle. They give us the very first and very last steps of making and secreting a protein. Our job is to figure out the 4 steps that happen in between. This is all about understanding the cell's production line, from blueprint to shipping the final product out the door. Okay, so the DNA blueprint is made, what's next? Well, that blueprint, the mRNA, has to get out of the nucleus. That's statement 2. Once it's out in the cytoplasm, the ribosomes jump on it and start building the protein. That's statement 3. After the protein is made, it gets wrapped up in a vesicle and shipped over to the Golgi body, which is like the cell's post office. That's statement 6. And what happens at the post office? It gets modified, folded, and packaged for final delivery. That's statement 1. So the correct order is 2, then 3, then 6, then 1. That sequence matches option B perfectly.
Right. Question 6 is a compare and contrast question. It asks how many types of structures with a double membrane that are found in animal cells are also found in plant cells. The keywords here are double membrane and found in both. So let's put on our thinking caps.
This one's actually pretty straightforward. Let's list the organelles with double membranes. We've got the nucleus, mitochondria, and chloroplasts. Now, which of these are in both animal and plant cells? Well, the nucleus is in both, for sure. Mitochondria? Yep, both need powerhouses. What about chloroplasts? Ah, that's the trick. They're only in plants. So that leaves us with two shared structures, the nucleus and mitochondria. The answer is B. Easy peasy.
Let's move on to question 7, where we're identifying a mystery molecule. We're told it's a polysaccharide, and we get 3 crucial clues. It's made in a plant, it's branched, and it's built from alpha-glucose units. Time to do some detective work.
This is a perfect time to use the process of elimination. Let's take the clues one by one. First, it's made of alpha-glucose. That immediately gets rid of cellulose, which is made of beta-glucose. Second, it's made in a plant. That rules out glycogen, because that's how animals store glucose. And third, it's branched. That lets us cross amylose off the list since it's an unbranched chain. So what's the only molecule left that fits all 3 descriptions? Amylopectin. That makes our answer A.
Question 8 is all about interpreting data. We see a picture of a triglyceride, and we need to match its 3 fatty acid tails to one of the 4 scatter plots. The plot shows the number of carbon atoms on one axis and the number of carbon-carbon double bonds on the other. Let's get to it. So all you have to do is carefully count what's in those 3 fatty acid chains. Let's do it. The top chain has 16 carbons and 0 double bonds. The middle one has 18 carbons and 1 double bond, and the bottom one has 18 carbons and 2 double bonds. So we're looking for a graph with 3 points, 16, 0, 18, 1, and 18, 2. Now we just scan the options, and look at that. Scatter plot B has those 3 exact points. It's a perfect match.
Okay, question 9. This one's all about what makes cellulose so strong. The diagram is showing us hydrogen bonds that are forming within a single cellulose molecule. It's not bonding to its neighbors, it's bonding with itself to stay straight and rigid. The question is, how does this specific feature help its function?
The key thing to notice here is that these are intramolecular bonds, bonds within the molecule. Think of it like a steel rod. These internal bonds are what keep it from bending or coiling up. They force the whole chain to stay long, straight, and rigid. And that straightness is a huge part of what gives cellulose its amazing tensile strength. The other options talk about linking different molecules together, but that's not what the picture is showing us. So the only correct explanation here is A.
And here we are, the last one for this part, question 10. It's a fun little logic puzzle. We've got 2 mystery solutions and a table of food test results. We know one bottle has starch and sucrose, and the other has glucose and protein. Our job is to figure out which is which.
Let's just walk through option C and see if it fits. If solution 1 is starch and sucrose, what do we expect? Well, a negative Benedict's test at first, because sucrose is a non-reducing sugar. But it should turn positive after we add acid and heat to break it down. And it should be positive for iodine because of the starch. Does Rho-Seal match? Yes, exactly. Now for solution 2 with glucose and protein. We'd need a positive Benedict's test for the glucose and a positive Biuret test for the protein. Does Rho-C match that too? It sure does. It's a perfect fit. This question is all about knowing your food tests cold.
And just like that, we've powered through the first 10 questions. But take a second and look at all the skills you've just put into practice. You've done magnification calculations, you've linked an organelle's structure to its function, you've sequenced a complex process, you've interpreted diagrams and data, and you've used the process of elimination. These aren't just facts, these are the core skills that will get you top marks.
You've all done some incredible work today, but this is just the beginning. To keep getting these expert tips, analysis, and loads more practice, you have to follow the one and only Mr. Chi everywhere. Seriously, find him on Instagram, TikTok, Facebook, and YouTube. He is the ultimate resource for acing this course. And of course, don't forget to like, subscribe, and share.
So what's coming up next? Oh, just the incredible world of enzymes and the mysteries of the cell membrane. Get ready because in part 2, we're taking on questions 11 through 20. Make sure you're subscribed so you don't miss it. I'll see you in the next explainer.